everyone, welcome to the NPTEL course named Experimental Biochemistry. This is week 11 and I am Beas Mukherjee, Prime Minister Research Scholar from the IIT Kharagpur, and today I will be talking with you about the protein ligand interactions. So this NPTEL course is coordinated by Professor Shagata Dasgupta and Professor Shomho Dev from IIT Kharagpur, and in today's lecture we will be talking about different kinds of protein ligand interactions that happen in nature. We will see what are the different applications of knowing about these protein ligand interactions. So in which all disciplines can you use this knowledge, and then we will also see what are the different biophysical and biochemical techniques using which you can know about these protein ligand interactions. Okay, so let us begin with the presentation. Right, so protein ligand interactions. Proteins, I am sure you remember, they are biomolecules. They are very large biomolecules which are present in our body, and most of the functions in our body are done by proteins. Now, who are ligands? Ligands can be small molecules. Ligands can be your DNA, RNA. It can be lipids. Okay, so it can also be another protein. So there is this interaction between a protein and its ligand. So ligand can even be a metal ion. Okay, it can even be your organic molecules. So protein ligand interactions refer to the binding interaction between a protein molecule and a small molecule known as ligand. In biological systems, proteins often interact with various ligands, which can include small molecules, ions, drugs, or other proteins. So these interactions play a crucial role in numerous biological processes, including signal transduction, enzymatic reactions, transport of molecules across the cell membrane, and regulation of gene expression. Right. So the binding of a ligand to a protein typically occurs at specific sites on the protein, and these surfaces are called binding sites or the active sites. These binding sites are complementary in shape and size to the chemical properties of the ligands. Okay. And that is why the ligands can bind to them. So now see, let us look at these interactions in more details. So there is specific recognition and binding between the ligand and the protein molecule. The binding process is often characterized by high affinity and specificity, with the protein binding tightly to its ligand under physiological conditions. So why do we study protein ligand interactions? So these are the different you can see the different fields of science in which protein ligand interactions find applications. Okay, so for example, there is drug discovery. So in drug discovery, you can find out the specific interactions of a protein, let's say a receptor or an enzyme, with the small molecule, which is the drug. Okay, so application is efficient drug design and lead optimization, pharmacophore modeling. All of these different applications can be carried out. What about structural biology? So our lab works with structural biology. Here what we do is we do NMR spectroscopy. So determination of three-dimensional structures of protein ligand complexes, that can be done using X-ray crystallography, it can be done using NMR, it can be done using cryo-electron microscopy. So that helps again for drug design, for understanding the mechanisms, molecular mechanisms of interaction. Okay, in enzymology, protein ligand interaction, this can be between the substrate, it can be between effector molecule, which can be an activator, it can be a repressor, that you can study. Okay, it can be between the your metal ions, it can be between small organic groups, cofactors and coenzymes. Okay, those and then after that, knowing about that, you can module this drug interaction, you can modulate, and that is how you can get drug enzyme interactions, you can get different metabolic engineering. All of that you can use this for. Okay, what about molecular modeling? This is a computational technique where you can actually design novel interactions between proteins and small molecules. You can dock different proteins and small molecule structures and you can analyze them. There can be molecular dynamics simulation. This also we do in our lab. Okay, so finally, binding mode prediction you can do. You can learn about the affinity of binding, the specificity of the binding using these protein ligand interaction computational tools. And then there can be virtual screening of ligand protein binding affinity prediction. All of that can be done. Okay, so now what other applications are there? So there are a lot of applications. Okay, so other applications include biomedical research. In biomedical research you can learn about your disease modeling and biomarker discovery, personalized medicine. So personalized medicine, I think we were talking about this in the last class. This is a very interesting concept. So in personalized medicine, what happens? Let's say a particular person is having a particular disease which is because of a specific, let's say, gene mutation. Then what happens? The entire genetic makeup of that person is taken into consideration, and according to that you are given a therapeutic. So even mRNA is very useful in these personalized medicines. Okay, so this is how you can treat patients. And for example, now when you go to a doctor, you are given one general medicine, right? But there it is not going to be general. It is going to be very, very specific to your genetic makeup, to your genetic mutations, and all of that can be screened, all of that can be analyzed, and a specific medicine that is going to be effective only for you is going to be given to you. Okay, and then chemical biology. Chemical biology, once again, I'm sure you understand the drug interactions, chemical genetics, pathway elucidation, all of these can be done. And then biophysical techniques. So this is what we are going to study about today. So in order to characterize the protein ligand interactions, we need a lot of biochemical techniques such as ITC, SPR, fluorescence anisotropy, and so on. Okay, so using these you can find out about the kinetics of the interaction, how fast it is happening. You can know about the affinities, that is how tight the interaction, how tight the binding is, and specificity, that is exactly who it is going to bind with, all of these things. Fine. So now we will move on to protein ligand interaction. So what exactly happens in case of protein ligand interactions, that you can see from this figure. Let's say this is an antigen antibody interaction, which is a very common protein interaction in our body, right? So how does this interaction happen? So there are these bonds, let's say hydrogen bond, there can be ionic bond, there can be hydrophobic interactions. Okay, so there can be hydrophobic interactions, there can be van der Waals interaction, ionic bonds. All of these different interactions are prevalent, and then protein ligand interactions can happen through all of these different chemical interactions between the protein and the ligand molecule. Okay, so now what are the different protein ligand interactions that we see around us every single day? So let us look at that. The connection is lost. Okay, no problem. So yes, so the different protein ligand interactions in our everyday life. Okay, so let's look at them. We already talked about these enzyme substrate interactions, right? These are very famous protein ligand interactions in our everyday life. Example, it's given your lactase enzyme, which is going to act on lactose and break it down to glucose and galactose. Then there is receptor ligand interaction. So in receptor ligand interaction, we have the cell surface receptor to which the hormones are coming and binding. Okay, for example, insulin binds to the insulin receptor on the cell membrane and initiates the cellular uptake of glucose. Fine. There is transport protein substrate. So here what happens, transport proteins bind to specific molecules for their transport across the cell membranes, and hemoglobin binds to oxygen in the lungs and transports it to tissues throughout the body. Fine. Then there is antigen-antibody interaction. The example of antigen-antibody interaction, let's say there are some viral protein and it is present as an antigen on the viral surface, and your antibodies produced by B cell are going to come and bind to these viral surface proteins, marking them for destruction for the immune response. Fine. Then there is ion channel ligand. So acetylcholine, this is binding to nicotinic acetylcholine receptors and allowing muscle contraction. Fine. So what are the other interactions? Let us have a look. Now this is very interesting. So where exactly do you see protein ligand interactions? We see it throughout the body. We see it everywhere in nature. Okay, so these three slides actually sums up some very common places where you see protein ligand interactions. This is a very interesting topic, and I would request you to read more about it, read more about protein ligand interactions and what are the different applications and how you can study them. So knowing about protein ligand interactions can make you, I mean, do a lot of things in a lot of different disciplines of science. Okay, so what are the other interactions? There is transcription factor DNA interaction. This we work with in our lab. Okay, so an example they have given, it is estrogen receptor binds to estrogen response element in DNA, controlling the transcription of genes involved in growth and development. Fine. Then there is coenzyme and enzyme interaction. So I told you about this. Enzyme binds to the see, ATP is binding to kinases, providing the phosphate group needed for phosphorylation reactions. Then there is plant pollination. So floral protein bind to the receptor, guiding them for plant flower pollination. Then there is microbial signaling. This is a very interesting topic. Bacterial quorum sensing. I'm sure you know about this. So quorum sensing is what? It is basically when a lot of bacteria are present together, they come to know about the stress condition or something in that place, because one bacteria releases signal and that is how they communicate. Okay, so there are receptors that bind molecules for quorum sensing on the bacteria, coordinating gene expression and behavior in response to population density. Then there is toxin, antitoxin. So snake venom and the snake venom toxin and the antitoxin to okay, so snake venom toxin binds to the ion channels in enzymes, ion channels or enzymes in the prey organism, causing paralysis and death. That is very scary, right? So there are other interactions as well, that is hormone receptor interaction. We talked about insulin, right? So thyroid hormone they are referring to here, and then neurotransmitter and neuronal receptor, that also we talked about. Again, there can be, these are again repetitive. Finally, we come to lectin and carbohydrate interaction. So lectin is a carbohydrate binding protein. So lectin carbohydrate interaction is important in our immunology, because if you remember the complement system is having these interaction. Concavalin A plant lectin binds to glucose and mannose residues on the cell surface, facilitating cell adhesion and agglutination. So drug natural target interactions, again morphine is there, which is a drug and natural target. These are the opioid receptors in the brain. So it is a natural painkiller, morphine. Fine. So next we move on to the molecular basis of the protein ligand interactions. So what are the molecular bases? So protein and ligands interact through a process known as molecular recognition. Molecular recognition involves specific and reversible binding between a protein, that is receptor, and a ligand, which is a small molecule or another protein, the nucleic acids, lipids, etc. This interaction is crucial for various biological processes such as signal transduction, enzyme catalysis, and so on. So what are the different characteristics that we need to keep in mind when we are studying about the protein ligand interactions? They are firstly the binding site. So binding site has to be complementary to the chemical groups on the ligand, right? That is one thing. Then the orientation, so binding site or active site, if you see, these sites typically have a shape and environment, right, complementary. Then there is complementarity. So protein binding site must have the orientation of the chemical groups, and the charge on the chemical group should be exactly complementary to those on the ligand surface. So therefore there should be complementary interactions. Then there is non-covalent forces. So most protein ligand interactions, these are not for life, right? So that means the bond, it is not supposed to be so strong that it would never dissociate, because binding and opening up, both of these happen maybe within milliseconds time scale, within let's say nanosecond time scale. It is that time scale within which it happens, okay? And therefore both the attachment and the detachment is necessary for its proper function. So if it is, let's say it's a transcription factor and it remains attached to the DNA, then what happens, that particular gene keeps on getting expressed, and if something is going it's not following the laws of nature, then we get cancer, right? So if the transcription factor is super strong, it is definitely going to cause problems in your body. That is why both binding and detaching is necessary, and that is why the forces are non-covalent here. Fine. So let us look at the other characteristics. There is specificity. So protein ligand interactions are mostly very specific. So specificity, once again, if you remember, the binding groups should be exactly complementary. The order, the arrangement, the orientation, everything should be complementary. Then there is binding affinity. So when we talk about binding affinity, it refers to the strength of interaction between a protein and its ligand. So the binding affinity, it can be nanomolar, it can be micromolar, it can be millimolar. Different ranges of affinity are possible. Induced fit. So what happens, let's say this is the receptor molecule, and initially this is the receptor molecule and this is the ligand molecule. So when it comes, the receptor molecule changes its shape to fit the ligand into its binding site. There is a conformational switch. So that is the induced fit mechanism. And then kinetics. Kinetics is how fast the binding is happening. After that, what happens? That entire thing, so binding and dissociation together makes the kinetics. Okay. What else do you have to know about protein ligand interaction? Nothing more. So now we will be looking at the different biophysical techniques that are employed to understand protein ligand interactions. So X-ray crystallography. This is a very powerful technique. So using X-ray crystallography, what can you do? You can understand the three-dimensional structure of a protein ligand complex, and you can also deduce the spatial arrangement of the atoms in the complex, providing details about the binding mode, about the interactions between the protein and the ligand. So this tells you exactly what kind of chemical interaction, what kind of chemical bonds are formed. This tells you what what is the conformational state of that complex of the receptor at that time, every single thing. Fine. Then we have NMR spectroscopy. NMR spectroscopy, you can perform HSQC, and from there you can do a titration experiment. So to the ligand, let's say to the receptor, you keep on adding the ligand. Then what happens? Then you get this chemical shift perturbations, and then from there you can find out that these particular residues are actually interacting with the ligand molecules. This is how you can do. So okay, so nuclear magnetic resonance, I'm sure you've understood. Then there is surface plasmon resonance. So surface plasmon resonance, ITC, these have been explained in details as well. Okay, so we will learn about these in details. So see, SPR is a label-free technique used to study biomolecular interactions in real time. So surface plasmon resonance is a very interesting technique and a very sensitive technique. So see, here what happens, there is a binding partner, typically a protein, and it is immobilized on a sensor surface. There is a surface which is gold plated, on that your protein is sitting. And then there is a tangential flow of the ligand. So ligand flows in, and what happens, initially when light was coming and going, it was reflected by the plasmon. So plasmons are nothing but actually free electrons that are present on the gold plated surface. So what happens, light, there was a specific refractive index, but then on protein immobilization, the refractive index has changed. Plus when the protein is binding to the flowing ligand, its refractive index is again going to change. So this change in refractive index is due to the formation of the complex, and that is monitored to understand the binding affinity, specificity, and the binding kinetics. Okay, another technique is ITC, which is isothermal titration calorimetry. Here what happens, in this technique you measure the amount of heat released during a biomolecular reaction. So by titrating a solution of protein with a ligand and measuring the resulting heat changes, researchers can determine thermodynamic parameters such as binding affinity, stoichiometry, and enthalpy of binding. So this is all about SPR. So yes, so this is all about SPR. You can see that there is this sensor chip. It is present on the glass slide, and on the sensor chip you are having gold. So there is tangential flow. This is tangential, right? So tangential flow of the ligand molecules, and on the sensor chip you have kept your protein. And then under that is the prism. The light is going, incident light, reflected light is going, and the refractive index you can find out from there. So SPR angle that is detected, and the angle of reflection and the intensity, both of these are plotted. So angle of reflection and intensity have been plotted. The principle, this is the entire principle of your surface plasmon resonance. So what about the objective? So surface plasmon resonance helps to perform interactions in real time. These studies can be used to observe if two or more interactants bind to each other. It can also be used to measure how strong the interactions are, and even measure the actual association and dissociation rate, specificity, and affinity of the interaction. So the objectives of this experiment are to perform protein-protein interaction using a surface plasmon resonance, to analyze the molecular interactions obtained using an SPR curve as a sensogram. So this is your SPR curve as a sensogram to calculate the binding affinity of the proteins interactions performed. Fine. We'll go to the next slide. So if you want to know more about the details about SPR, you can actually refer to this slide. If you don't want, we can skip to the next slide. So for your interest, I will go through this. So surface plasmon resonance is a phenomena that can be, one second. Okay. So surface plasma resonance is a phenomenon that can be observed in thin conducting films at the interface between the media of different refractive indices. Okay, so in SPR system, the media are of glass, media are the glass of the sensor chip and the sample solution, and the conducting film is a thin layer of gold on the sensor chip. The SPR is a physical process that can occur when plane polarized light hits a thin metal film under total internal reflection conditions. So when a beam of light hits a half circular prism, the light is bent towards the plane of the interface when it is passing from a denser media to a less dense one. Fine. So then what happens? Denser media to the rarer medium. So changing the incidence angle changes the outcoming light until it reaches a critical angle. At this point, all the incoming light gets reflected. So that is your total internal reflection, right? So because you are going from a denser media to rarer media and it is beyond the critical angle or IC, then what happens? There is total internal reflection. So this is completely going into the denser media again. So the surface is coated with a thin film of noble metal, usually gold. Gold is used in the sensor chips because it combines favorable SPR characteristics with stability and high level of inertness in the biomolecular interaction context. At a certain combination of angle of incidence and the energy or wavelength, the incident light excites plasmons. Plasmons are the electron charge density waves in the gold films, and as a result, a characteristic absorption of the energy via the evanescent wave field occurs, and SPR is seen as a drop in the intensity. So there is a drop in the intensity of the reflected light as we see in the previous figure. So in an XPR experiment, one molecule, that is a ligand, is immobilized on the sensor chip, and the second molecule is the analyte, which is flowing through, right? And then you measured the response in response units, and it is proportional to the mass on the surface for a given interactant. So the response is proportional to the number of molecules bound to the surface. Response is recorded and displayed as a sensogram in real time. So I hope that is clear. Okay. So hopefully that is clear. Now we will move on to the next technique, which is isothermal titration calorimetry. So we will look at this. Let's see what happens in ITC. So ITC is a technique. Okay, so I already told you that here what is measured, the amount of energy that is given out during a chemical reaction, that is measured. So the objective of ITC is that ITC is one of the very important techniques used to determine label-free specific interactions between molecules dissolved in an aqueous solution, such as protein-ligand binding, protein-protein binding, DNA-protein binding, protein carbohydrate binding, protein lipid binding, antigen antibody interaction, and so on. So it is a highly sensitive techniques that works by titrating one binding reagent, titrant, generally a small chemical compound called a ligand, into the titrant. So there is, once again, there is a titration experiment. So what happens, the protein of interest is there, and into the protein of interest you add the titrant, okay, which is your ligand. So you are adding the titrant into protein of interest. Then what happens? This technique is commonly used for drug development as it provides valuable thermodynamic information about the interaction of the molecule with its target protein. ITC can precisely determine Gibbs free energy, enthalpy, entropy, heat capacity changes associated with the binding and interaction of the biomolecule. So this is a very, very sensitive technique. Fine. Now to understand ITC better, we have this entire explanation. So look at this. The ITC instrument measures the amount of power in microcalories per second required to maintain a constant temperature difference close to zero between the sample and the reference cell. So if you remember the reference cell, this was your reference cell and here was your sample cell. So you need to find out the temperature difference between the two. Fine. So what happens here? Each injection of the serine solution, usually termed as ligand, triggers the binding reaction, and depending on the binding affinity, the concentration of the reactants, that is macromolecule M and ligand L in the cell, a certain amount of macromolecule or ligand complex is formed. The formation of the complex is accompanied by the release or absorption in case of exothermic or endothermic reactions of heat that causes a difference in temperature between the two cells. So then the feedback system either lowers or raises the thermal power applied to compensate for such temperature unbalance. So what happens here is, let's say that you have titrated the ligand into the protein. If it is an exothermic reaction, the temperature is going to increase. If the temperature increases, the feedback system is going to lower it. Otherwise, if it is not an exothermic, it is an endothermic reaction, your temperature is going to get absorbed by this absorbed from the system, so it lowers, and the feedback system will raise the temperature. So this action of the feedback system is actually recorded by the instrument. Then what happens? See, after injection, the system reaches equilibrium and the temperature balance is restored. Therefore, the recorded signal shows a typical deflection pattern in the formation of a peak. Integration of the area under the peak, assuming that the baseline as reference provides the amount of heat associated with the injection. As the reactant in the cell becomes saturated, the heat signal diminishes only until background heat is present. In a properly designed typical ITC experiment, the binding affinity or dissociation constant, or KD, okay, or the binding constant. So binding constant is your Ka or Kb, which is 1 by Kd. So the stoichiometry can be known, enthalpy of the protein can also be known at a constant temperature, Gibbs free energy can be known, entropy, etc. Everything can be calculated. Fine. So if one protein molecule M binds to the ligand, then the one-on-one binding model is applied. So this is the graph. You can see the power, you can see the time, and this tells you that whatever interactions have happened. Okay. So then we will move on to the next slide. What about fluorescence anisotropy? So fluorescence anisotropy is a technique to study again protein ligand interactions. So here what happens? So okay. So in case of fluorescence anisotropy, we can see that this can be employed to study protein ligand interactions using fluorescently labeled proteins or ligands by monitoring changes in the fluorescence intensity, polarization, or a lifetime upon ligand binding. So researchers can determine binding affinity, kinetics, and conformational changes in the protein. So what happens in fluorescence anisotropy is that when uh, polarized light hits a compound, okay, then what happens if the compound is very slow moving? So for example, it is a protein ligand complex. The complex is going to be very big, so it is going to be very slow tumbling, so it is going to move slowly, and then what is going to happen? It is going to give back the plane polarized light as it had initially come. But for a molecule that tumbles very fast, here what happens? The plane polarized light gets depolarized. So plane polarized means the electric and the magnetic field vectors are like vibrating along a specific direction. If this is electric field vector, this is a magnetic field vector. They are oscillating against a specific single direction. That is what plane polarization is. But then depolarized meaning they are going to vibrate in along different planes. So the principle of using fluorescence anisotropy is to measure protein ligand interactions. Free fluorescent ligands or peptides irradiated with plane polarized lights quickly rotate and emit light in a depolarized manner. So fluorescent ligands bound to larger proteins tumble slowly and emit polarized light. The degree of molecular binding can be inferred from the amount of polarized light emitted. Fine. So now we will see the different. There is one more technique left, which is your EMSA. This is something that we do in the lab almost every single day, and after that we will go for the question answers. So in EMSA what happens, you can see this in EMSA. One protein is binding to your nucleic acid. Here it is RNA. That is why I refer to this as R. Okay. So protein is binding to RNA. First, the binding kinetics is given by K on, and the dissociation kinetics is given by K off. So this is your rate constant. So rate constant for binding, it is K off. Rate constant for dissociation, it is binding is K on, and for dissociation it is K off. Alright. So now if you see, the total protein is equal to amount of free protein, the one that has not bound to the ligand, plus the amount that has been bound to the ligand. Okay. So R total is once again free RNA and bound RNA. So the fraction bound, that is equal to protein into your RNA divided by free RNA plus protein into RNA. So that is complex by free RNA plus complex. That is the fraction bound. Again, fraction bound is equal to free protein divided by free protein plus Kd. This comes together. Now free protein you can not measure, because this free protein, exactly after adding the RNA, exactly how much protein is free, how much is bound, that we cannot measure externally. What we can measure is total protein, right? And therefore we consider this entire complex as the P total divided by P total plus Kd. So that is an assumption. Okay. Then after that what we do is, so initially we start with very low or negligible concentration with okay, so here actually the concentration of the protein is much more than the concentration of the ligand, and that is why we can assume that free protein is nearly equal to total protein, because most of the protein is free. So therefore your Kd is equal to free RNA into free protein divided by the complex. Fine. So we will move on to the assignment questions. Okay. So for the assignment, this is the first question. So question on protein ligand interactions. For a binding reaction, protein P and ligand L gives me PL. The unit of K on and K off have the units dash and dash respectively. You try to solve the question. I will give you the answer in 2-3 minutes. Right. So for this you can see what is the unit of K on and K off. Just recently we studied about these. So let us look at the answer. So the units of K on and K off are moles per second, and okay, so the one for K off is second inverse. So just to confirm, we can check here. Yes, so mole inverse, second inverse, and K off is your second inverse. Correct? Now, which of the following experiments can be used to determine the binding kinetics under favorable conditions? Okay. So, which of the following experiments can be used to determine the binding kinetics under favorable condition? One is EMSA, there is ITC, there is fluorescence anisotropy, and there is surface plasma resonance. So, which can tell you about the binding kinetics under favorable conditions. So yes, all of these answers are correct. All the options are correct because all of these can tell you about the binding affinity of a protein ligand interactions under suitable conditions. Right. Then the next question is, enthalpy of a binding reaction can be determined from. So enthalpy can be determined from ITC, fluorescence anisotropy, surface plasma resonance, and EMSA. So right answer should be ITC because we studied about this right very recently, that yes, ITC can be used to determine the enthalpy of a reaction. Then the next one is dissociation rate constant, which is K off for a protein DNA interaction. This is determined by what? EMSA, the intensity of the free ligand can be fit to which of the following equations to obtain K off. So how can you get the intensity? So of course, I is equal to I naught into 1. So which one should be the right answer? Let us see. And for the next question, which of the following statements about protein ligand binding is true? Ka, or the association constant, is independent of conditions such as salt concentration and pH. When all the binding sites are occupied, the Ka or association constant equals the ligand concentration. That is wrong because Ka is equal to protein concentration when half of the sites are occupied. So Ka or association constant is increased when the binding will be tighter. That is true. So the larger Ka or association constant means weaker affinity. So let's look at the answers. Okay. Yes. So this is your right answer, and again C is the right answer for this. For the next question, this is a graph given from ITC. What does this tell you about the total heat? So how can you find out the total heat? You will have to add all the peaks, right? So integration needs to be done. That is how you can find out. What about this question? And this is a very interesting question, and you can get these kind of questions in your competitive exams as well. So see, it's a match the following and titration, isothermal titration calorimetry. Which one is it going to be? It is used to detect physical technique to detect thermodynamic parameters of interaction. So your 1 should be 2. Fluorescence anisotropy, It is a phenomena where the light emitted by a fluorophore has unequal intensities along all axes of polarization. So your 2 should be 4. So 4 . Okay, yes. 2 is 4, correct. So that is either C or D. Now, between C and D, which one do you think would be the case? So surface plasmon resonance, this is the resonant oscillations of conduction electron at the interface between negative and positive permittivity in a material. So this should be your surface plasmon resonance. So 3 should be 1. So your D is the right answer because EMSA would definitely be the protein complexes with nucleic acid. This is what it is used to determine. So the right answer should be D. What about the next question? See, cytochrome P450 enzyme binds to a newly synthesized drug molecule with an association constant Ka is equal to 10 to the power 7 mole inverse. What is the most likely measured Kd in the presence of a competitive inhibitor? So they have given you Ka, you are asked to find out D. It's a very simple question. So Ka is equal to 1 by Kd, and therefore you can just get a reciprocal and you can find this out. Right. So what about the next one? Enough free energy del G for the binding interaction is known. Then how do we calculate the number 1 is association constant? Then we can calculate what association constant, dissociation constant, binding energy K on and dissociation energy K off, which all can be found out from free energy. So from free energy, which all can you find out? 1 and 2. You can find out about dissociation and association constant. Next we have the K on and K off values for ligand X and Y with the enzyme E. So X, ligand X is having K on 10 to the power 4 and K off 10 to the power minus 3. Ligand Y is having K on 10 to the power 7 and K off as 10 to the power minus 1. Which of the following statements is or are true? X binds to E more tightly. Y binds to E more tightly. This is definitely true. X-E complex is longer lived. So which one binds to which one more tightly and which one is longer lived? So for longer lived you will have to see this. Okay. This is going to give you the time information. So YE complex is longer lived than XE complex. So 4 is definitely correct. And X binds E more tightly, or your this one binds E more tightly. So let's look. So right answer is C, because this complex is more longer lived and more tightly who is binding, Y is binding to E more tightly. Correct. For the next question, ITC, for a titration experiment to determine the dissociation constant, it is necessary to vary the temperature. The ligand concentration is varied and the protein concentration is kept constant. That is true. The protein concentration is varied and the ligand is kept constant. The concentration of the free ligand, free protein bound complex must be determined. Okay. So, which of these is true? It is 2 and 3 are correct. right. Then for the next question, the researchers designed a new drug molecule and wanted to study its interaction. For this drug molecule, so with human peptidase enzyme, if the peptidase drug molecule complex dissociates with an off rate of 1549 per second, then what is the time constant tau for this reaction? You try to solve this. I will get back in 5 minutes. Right, so we were looking at this question. Let's see how to solve this. Okay, so your tau is equal to 1 by k of, and if you do the calculation, you get 645.5 microseconds as the answer. Okay, so that is your time constant for the reaction. Then there is another question. This is the second last question for today. Try to solve this. The dissociation rate constant here for a protein-DNA interaction is determined by EMSA. The intensity of the free ligand determined at a corresponding time interval is given in the table below. Determine the K of M. So the intensity is given, time is given. You have to plot it and then you determine the K of. So if you plot it, you can find out the K of value from here. Okay. So intensity and time for EMSA is given. This is another question. Right. So this is another question where they are asking very similar protein ligand titration experiment was repeated where the protein concentration of 10 micromolar is used, which is more than the expected Kd, and the concentration of the ligand for the protein ligand complex for each titration point is given below. Determine the dissociation constant. So ligand is given protein ligand concentration you are getting in micromoles, and the ligand concentration is again separately given, PL concentration you are getting. So from here you have to find out the dissociation constant. So how do you find out the dissociation constant? So ligand concentration, we can see here it is gradually getting increased, and protein ligand concentration is also increasing, right? And the total protein concentration is given to you as 10 micromolar. So if you remember the calculation for EMSA, we learnt about EMSA calculation here. This slide you have to refer to for this. Okay, and if you refer to this slide, you will be able to do the calculation. Right? So, and by doing the calculation, you can get the right answer as 1 micromolar. Okay? Thank you for attending my class. These are the different websites, the different papers that you can refer to, different books, and thank you for attending the class. So I will be ending the class here.